Hello, we are the CPU Gliofighter Group. Synthetic biology is a multidisciplinary field of study that involves the design and construction of new biological components, functions, and systems to build controlled ways, biological logic, and production systems that do not exist in nature. Synthetic biology can be applied in many fields, of which healthcare is its first application market. As students of medicine, we are always concerned about the development of the world pharmaceutical industry. So, with the glioma situation so dire, we developed a Hepato Stella System. Nowadays, the treatment of glioblastoma has been paid more and more attention by the society, and it is a difficult problem to be solved. The standard treatment for newly diagnosed glioblastoma (GBM) is surgical resection followed by radiation therapy, supplemented by multimodal temozolomide. At present, the standard treatment for GBM is called STOP protocol, which includes as complete surgical resection as possible, followed by fractional radiotherapy of 60 GY, combined with synchronous temozolomide chemotherapy, followed by 6 months of adjuvant chemotherapy. This treatment regimen can only gradually extend the median survival of patients from 2.5 months to 14.6 months. Recurrent brain gliomas are almost universal, and treatment options for recurrent GBM include surgical resection, directed radiation therapy (SRS), systemic application of the DNA alylation agent lomustine, and systemic application of the anti-angiogenic monoclonal antibody bevacizumab. Each of these therapies has shown some efficacy against recurrent tumors, although the efficacy is limited and inconsistent. Not all are associated with severe treatment-related toxicity. Also, many drugs have difficulty crossing the blood-brain barrier. To effectively address the challenges in the treatment of glioblastoma multiforme (GBM), we hope that RNA interference (RNAi), enabled by small interfering RNA cRNA, will serve as an emerging therapeutic strategy. This method can accurately regulate tumor genes at the level of messenger RNA, mRNA, or protein, and has higher specificity and therapeutic efficiency compared with traditional chemotherapy. However, the rollout of cRNA therapy faces some significant challenges. First of all, naked cRNA has low stability and is easily degraded by nucleases in serum, thus reducing its effectiveness in vivo. Secondly, the physical properties of cRNA make it difficult to penetrate the blood-brain barrier (BBB) and the cell membrane of target cells, which limits the entry of most therapeutic cRNA into target cells in the brain for effective treatment. To address these issues, in vivo cRNA delivery is primarily achieved through cationic charged nanoparticle delivery systems, including lipids, inorganic nanoparticles, and mixtures. In the context of our proposal, we mainly selected lipid nanoparticles that have been widely used in cRNA delivery studies. Due to their biocompatibility and ability to efficiently package cRNA, we compared the human body to a vast galaxy, and the different organs and cells in the human body are compared to a planetary body. Exosomes play a huge role as rocket ships for communication between planetary bodies. HSS can modify the endogenous cells of the human liver, regionally an ordinary planetary body, transforming them into galactic factory cells, or factories that can produce specific live rockets. The essence of this live rocket is exosomes, but the specific live rocket produced by the galaxy factory cells can wrap the exogenous cosmic heavy elements, that is, the drugs needed for brain glioma treatment, and better achieve astral development, that is, the treatment of human brain glioma. Galaxy factory cells selectively deliver and package an exogenous heavy element of specific genes, called cRNA, into live rockets via nanoliposal gleam carrier particles and LGCP. Live rockets encased in genetic heavy elements can be launched using the galaxy's own circulatory system to target the delivery of genetic heavy elements to specific stars' brain gliomas. At the beginning of the design, HSS can be organically genetically modified to assemble instructions into the system, so that the live rocket launched by it can have a starlight navigation tool targeting protein. That is, a starlight navigation tool is embedded in the shell of the live rocket, and this specific tool can be accurately navigated and can be combined with a specific target on the blood-brain barrier in the brain to guide the rocket of life through the blood-brain barrier smoothly into the brain. Then, the star navigation tool accurately locates the live rocket to a specific star brain glioma cells. The live rocket shell breaks, and the gene-heavy element will automatically release to find the matching target gene, so that the gene sequence that promotes the growth of brain glioma is destroyed by the gene-heavy element, interfering with the expression of the gene, so as to achieve the targeted treatment of brain glioma. HSS is constructed in vitro, and the backbone part is an organic molecule RNA sequence. Based on this sequence, we use organic gene modification technology to modify the sequence, and we can insert the starlight information molecules we need. For example, the navigation tool described above is edited and synthesized by the inserted sequence. In order to realize the function of the sequence, we put a starting sequence in the front end of the sequence as an activation signal, and after the sequence reaches the galaxy factory cell, the activation signal will first play a role, so that the whole sequence will wake up from the sleep and play a role. The synthetic system is composed of 3 modules. Through the synergistic effect of high-precision genetic modification and activation signal, individual customization can be realized, and industrial production can be carried out conveniently and efficiently. It can ensure the stability of the production chain, significantly reduce the off-target effect, and ensure the specificity and safety of the system. The question of how HSS travels from in vitro to deliver in vivo the glycogen factory cells is also an important one, for which we use bioinformation carrier technology to wrap an LGCP, a nanoparticle lipid particle, in which organic RNA sequences bound to the heavy elements of genes are wrapped as bioinformation carriers. These carrier particles are highly biocompatible and targeted, and can transmit core information stably in vivo. We can administer the drug intravenously, inject the wrapped core sequence into the body, and then the sequence and its contents will travel through the body's blood circulation system to the liver, where they can be used in stem cells. Regarding our lipid nanoparticles, their main role is to ensure that the core sequence is not degraded by the human body before entering the galactic factory cell. This design not only optimizes the transmission efficiency, but also minimizes interference with the human body. Because of the autonomy, safety and efficiency of the HSS system, we can quickly change the treatment strategy at any time according to the patient's situation, and achieve the effect of one person, one therapy to defeat the enemy. To demonstrate the effectiveness of the system, We first performed functional validation in vitro. The experimental cells prepared included HEC290, 3T and U80, 7mg cells. We input the complete system into the experimental cells and detect the protein expression of the silent target 24 hours later. By detecting the expression of the corresponding protein, we can confirm whether the system has played a role in the cells. At this time, HEC290, 3T cells are both chassis cells and target cells. After co-culture with HEC290, 3T cells for 48 hours, cell culture medium was collected from HEC290, 3T cells, and GMV produced by the cells was obtained by high-speed centrifugation, and then purified and enriched GMV was obtained by reinserting exosomes in PBS. The physical characteristics, shape, and size of the collected GMV can be observed under transmission electron microscopy to confirm that GMV can be secreted normally, and the shape and size of GMV are not changed. The collected GMV was cultured with U80, 7mg cells, and the collected GMV was added according to the ratio of 1 to 10. After 48 hours of GMV and cell culture, the expression of U80, 7mg cell target protein was detected. In vivo functional study. Firstly, the system was injected into normal mice, and GMV was extracted from mouse plasma 48 hours later and purified for further analysis. First, whether the system directed the efficient loading of cRNA into GMV and the detection of cRNA in exosomes was checked. Fluorescence analysis. We used commercially available EGFP transgenic mice to verify that exosomes can reach the brain. First, cRNA targeting the brain with EGFP gene silencing was designed, and the fluorescence intensity of the mouse brain was analyzed to confirm that the exosomes reached the brain through the blood-brain barrier and interfered with the target. The system was injected into diseased mice, and in order to verify that the liver was the site of the initial assembly of exosomes, mouse primary hepatocytes were isolated at different time points after the injection of the loop into the mice, and cRNA levels in exosomes extracted from the primary hepatocyte culture were assessed at different time points. At the same time, major organs such as heart, kidney, liver and lung of mice were collected and cRNA concentration analysis was performed on the organs to ensure that exosomes reached these parts. To verify the therapeutic effect, first, the weight of the mice was recorded every day. Second, the expression of silencing protein and mRNA in animals. Third, analysis of diseased tissue sections. That's all. Thanks for your listening.